Hey everyone, we are continuing chapter 10. We are on slide 16. We just discussed the basic differences between muscle, cardiac muscle versus smooth muscle versus skeletal muscle. We're focusing now on skeletal muscle, the muscles that move your bones. We're going to look at the organization a lot here, both in terms of gross anatomy as well as tissue cellular level anatomy. When you look at a whole muscle is an organ. Like if you look at your biceps brachii, or in this picture, it looks like they're looking at the brachioradialis or something. When you look at a single muscle, that's an organ. A muscle is an organ. A whole muscle is an organ, just like your heart is an organ, just like your brain is an organ. A whole muscle is an organ. It's an organ because it has muscle tissue, it has epithelial tissue in certain parts, with blood vessels. It has nervous tissue in it because there's nervous signaling to it. It has connective tissues. It has all tissue types within that organ working together. There is, of course, muscle tissue within a whole muscle, but there's other tissues as well. Specifically, the connective tissues that are in a whole muscle, there are three names of how these connective tissues are organized. Three ways that connective tissue organizes whole muscle. Whole muscle, I'm going to go backwards a little bit here. Whole muscle is surrounded by epimysium. Epimysium is what is covering the outside of whole muscle. In this cross section up here, epimysium is just the outside. And in this simplified drawing over here, it's in blue, but I'm going to color over it in green. That's epimysium. Epi means on top or to the side. MYS, the mys part, is referring to muscle. It's the connective tissue covering muscle, epimysium. It is made of dense, regular connective tissue, surrounds the whole muscle. Within whole muscles, you've got bundles of cells. You've got a bundle shown here in our cross section, here and here and here. The word for this bundle is a fascicle. A fascicle means bundle. So a muscle fascicle is a bundle of muscle fibers, of all the cells of the skeletal muscle. What's surrounding each fascicle is perimysium. So everywhere in pink that I'm drawing, and also here in this simplified drawing to the right side, this is perimysium. Perimysium is also made of dense regular. Its location is just surrounding fascicles rather than surrounding the whole muscle. Finally, we have individual muscle fibers that are made into those bundles of fascicles. So if you take a look down here, an individual muscle fiber, there's several of them packed into here. In our cross section, they just look like little dots, but in actuality, they're very long. And once again, in our drawing over here, what I'm coloring in blue, I'm not going to do each one, but those are the muscle fibers. What surrounds muscle fibers, what covers and protects muscle fibers, which I'm going to color in orange here, what covers and protects muscle fibers, that's endomysium. You've seen endo before. Endo means within. It's within the muscle. Endomysium is a connective tissue that surrounds individual muscle fibers. So we've got several levels of organization here. Cellular level muscle fibers surrounded by endomysium. You bundle a bunch of muscle fibers together into a fascicle. That fascicle is covered by perimysium. bundle a bunch of fascicles together, you get whole muscle. Whole muscle is covered by epimysium, and they're all made of dense regular connective tissue. Now, if they're all made of dense regular connective tissue, are they at all related? Are they at all connected? Take a look over here at the end of the muscle. If muscle is lengthwise attaching to something, we need connective tissue to connect our muscle to bone. That's where tendons meet. Tendons connect muscle to bone, and it's the convergence of those three connective tissues, epimysium, perimysium, and endomysium. It forms a firm attachment between the muscle and with the bone. And that tendon is not only meshed in with the muscle fibers and fascicles, but it's also forming the periosteum of bone, that outer layer of periosteum, not the osteogenic layer with the osteoblasts and osteoclasts, but the outer layer, the superficial layer that has the periosteum that has is a continuous with tendons. It's made of dense regular as well. You can see in this histology on the left side, muscle is over here. The dense regular connective tissue of tendons running in this direction is here. The muscle here is in cross section. You can see circular shapes. Over here, on the right side, this is the muscle. Muscle fibers are cut in longitudinal section. And then over here, you have the tendon. Just two different views, different dyes. You can see they're stained differently, but it's the same. We're looking at the same thing. That's how muscles are organized in a gross anatomy histology level, tissue level. And one special kind of tendon, let me go back. The definition of a tendon is connecting muscle to bone, because after all, the goal of a skeletal muscle is to move bone, so we need to attach to bone. Sometimes though, tendons can stabilize muscles. Tendons can connect muscle to muscle. We have a special word for those type of tendons. A special tendon that connects muscle to other muscles, that's called an aponeurosis, plural aponeuroses. By that name, you might think it's nervous tissue, and that's what early scientists, people who had dissected bodies before, thought it was nervous tissue, but they're incorrect. Unfortunately, the name stuck, so we're stuck with this name for now. Aponeurosis is when you have a connection between two muscles, and it's tendon. So it's also dense regular. It's a thick, flattened sheet. There's aponeuroses in several places in your body. There's two that I want you to know. One is connecting the abdominal muscles. This is known as the abdominal aponeurosis. And then one connecting this muscle in the front of your forehead, and then this muscle in the back, the frontalis in the frontal region, and the occipitalis in the occipital region. Connecting the two on the top, that's the epicranial aponeurosis. Epi means on top. It's on top of the cranium. Epicranial aponeurosis. This secures these muscles together and also influences them. When one moves, it'll pull the other with it. Helps keep things tight and in line. All right, so we've seen that an individual muscle fiber is surrounded by endomysium. What we're drawing here is a muscle fiber in the longitudinal section. It is very long. On the histology we keep seeing, we can never show the full length of a muscle fiber because they go off the page. They can be up to 30 centimeters in length. I don't have a ruler next to me, but 30 centimeters can be like about a foot, maybe 12 inches or so. I can't get my conversions, my metric to standard units. I can't do that quickly, but it's long. It can be as long as your brachial region. It can be long as your femoral region. Those muscles are made up of muscle fibers that are really, really long because muscle cells, myoblasts, fused together. The nuclei tend to be pushed off to the side. You can see a nucleus here and a nucleus here. They're always pushed off to the edges. And remember, they're multinucleate. The term for the plasma membrane of a muscle fiber has a special name. It's called the sarcolemma. Sarco, whenever you see this prefix, it's referring to muscle. Sarco and myo, M-Y-O, both refer to muscle. Sarcolemma is referring to the plasma membrane of muscle. Why do we bother calling it something different? Because it's unique in some ways. We're going to see how it's unique coming up. Here's one example of how it's unique. It has extensions that go deeper into the cell. So let's look at this picture down here. You can see the sarcolemma is just all this outside stuff that's bordering the edges of the cell. We're only looking at a fraction of the cell. The cell is actually really big and really long. We can't see the whole thing right now, but we see the sarcolemma here. And just like if you were a gopher and you dug holes into the ground, from the surface into the ground, and you create this huge network of tunnels underground, those tunnels underground are still connected to the surface. Here, take a look at the bottom in green. Here we have a tunnel that goes deeper into the fiber and creates this crazy network of tubes that are still connected to the surface at multiple points. So there's a network of tubes that are connected and continuous with the surface of the sarcolemma. So it's made of membrane. It's just a weirdly extended branched network of membrane continuous with the plasma membrane, with the sarcolemma. The term for these structures, they're called transverse tubules. You can abbreviate them to T tubules. These are T tubules. And these T tubules, what they do is that they're increasing surface area. You're creating more space for membrane, but the function of this surface area is to better spread electricity. Spreading electricity deeper into the muscle fiber. Have you ever heated up leftover food? If it's really cold, you sometimes have to like stab holes in that food to make sure that when you heat it up, it more evenly gets to all the deeper parts. Because otherwise, if you just stick that frozen thing into the microwave, the outside will be nice and warm and cooked, but the inside will still be really cold. By having holes, not really holes, but tunnels into the fiber, you're spreading electricity better. You don't want just the outside of the muscle to contract. You want the whole muscle to contract evenly. That's the function of T tubules. Hope that weird metaphor made sense. We also give a name to the cytosol of the muscle cell. It's called the sarcoplasm. Again, sarco just means muscle. Plasm, it's referring to the stuff inside of the cell, sarcoplasm. This will be an important place. Like sarcoplasm is everything else, all that space in between here. There's other stuff here that we're going to talk about, but it's all the fluid inside of the cell. It'll be an important place for sending ions, sending electricity to cause contraction. So we've gone over the sarcolemma, T tubules, sarcoplasm. What are these other structures in here? These rod-like structures that are running longitudinally? These rod-like structures, they are called myofibrils. Myo means muscle, fibril means small fiber. So muscle fiber is the whole cell. Fibril are these really long protein things inside of the cell. So they are made of protein. These myofibrils are made of protein. We're going to go in a minute look more in depth of what they're made of. And they attach to the sarcolemma at each end of the muscle cell. We're going to see that these myofibrils are the ones that shorten somehow. And by attaching to either end of the fiber and shortening, you're pulling on whatever it is you're attached to. If you're attached to a bone, you're going to pull that bone and cause movement. That's how muscles, skeletal muscles, cause movement. They grab onto a bone and pull on it. They don't push. So myofibrils are the packed proteins all within a muscle fiber. All of these cylindrical rod-like shapes you see here. You'll notice that in blue, you'll see this network of membranous stuff surrounding each myofibril. And it's also shown here. This network of membranous stuff within the cell that's surrounding each myofibril, this is the sarcoplasmic reticulum. It should sound familiar. It sounds like endoplasmic reticulum. It's a specialized endoplasmic reticulum. Endoplasmic reticulum, if it was rough, it was good for making protein. If it was smooth, it was good for assembling lipids. Sarcoplasmic reticulum has a different function. Because it's specialized to the muscle, it stores and releases calcium ion. Calcium ion is really important because it helps signal contraction. We're going to see how in a minute. But we've seen calcium in bone before. Here's calcium in muscle for a completely different reason. We're not hardening this calcium. We're using this calcium as a signal molecule, signal ion. The sarcoplasmic reticulum surrounds all these myofibrils. So there's tons of them wrapping around each myofibril. Let me actually show you this real quick in Complete Anatomy. I have this linked in my videos section, but if you go to Complete Anatomy and go to models, at the bottom left, if you click on models, normally you would be set on female or male to look at the whole body, but if you click on detailed, one of the things you can choose, where'd it go, is skeletal muscle? Yeah, let's start with skeletal muscle first, and then we'll go to skeletal muscle fibers. So if I click on skeletal muscle, here you can see a piece of a whole muscle. You can see the tendon at the edge. What's surrounding the whole muscle is epimysium. If you look at an individual fascicle, surrounding that is perimysium. If you look around an individual fiber, that's endomysium. We're in longitudinal section right now. Oops. And this is in cross section. You can see the difference. Cross section, you see a bunch of circles. Longitudinal section, it's more rectangular because these are long fibers. So go back to an individual muscle fiber. So here's an individual fiber. Surrounding the whole fiber is endomysium. You can see that, are they going to show? I can click on the sarcolemma, which is just deep to the endomysium. It's highlighted in green right now. And then all these circular cylindrical things, those are all myofibrils. The things labeled in, the things in beige, colored in beige, those are the sarcoplasmic reticula. And they're wrapping around the myofibrils. The nuclei are pushed to the edge. It's these large purple things. Is there another one over here? There's one hiding under here. And then the blue things, there's lots of mitochondria. You need mitochondria to power these muscles. We're going to look in more detail Oh, one more thing I want to point out. Those sarcoplasmic reticula are bordered by T tubules. So T tubules are surrounding each sarcoplasmic reticulum on its edges. We're going to look closer at these myofibrils and learn why they have a striated appearance, a striped appearance. So we're going even deeper. We started with a whole muscle, to a fascicle, to a fiber, to a myofibril. I'm going even smaller here. So shown here is a single myofibril. It's not the full thing. It's going to be much longer in this direction. It's going to be much longer in this direction. We're just looking at a segment of it. And if you look at how it's arranged, you'll notice that there's these, what's colored in this diagram, darker purple, and then lighter pink. Now, I'm not doing this perfectly, but there's this alternation of darker purple and lighter pink over and over and over again. What's represented as the darker purple, those are called thick filaments because they're thicker and they're filament-like, they're long and thin. They are made of protein, and the main protein they're made of is called myosin. Myo means muscle. Anything that ends in IN usually is a protein. Myosin. The ones in pink, salmon color, that's actin. Actin is another protein. Proteins are really important for muscle, if you haven't heard that before. So you have actin proteins that form thin filaments, because they're thinner, and you have myosin protein that forms thick filaments. And they overlap over and over and over in this pattern. The term for a repeating unit, which includes the thick filaments in the middle, half of thin filaments on one side, half of thin filaments on the other side. The term for this is a sarcomere. So a sarcomere is a segment of a myofibril. You have repeating sarcomeres over and over and over again along the entire length, and they look exactly the same. So you can see another sarcomere here. And also not shown would be another here, and on and on and on. Repeating segments. Because of these repeating segments, you have areas that appear darker, where the thick filaments are, and then segments that appear lighter, where you only have thin filaments. Because of this pattern of overlapping actin and myosin, you get stripes, you get striations. And that's why we can see it with the histology of skeletal muscle, as well as cardiac muscle. They all have this pattern with their sarcomeres. Focusing on the thick filaments, so if we zoom in on this, what we see is that these thick filaments are made up of those myosin proteins, and an individual myosin protein looks something like this. It's got this long tail region. It's got this region that can bend. You're going to talk about that in more detail in physiology. The thing that I want you to pay attention to are these two head regions. These head regions can grab onto things. Specifically, what they can grab onto. Take a look back up over here. These myosin heads can grab on to the thin filament. The term for this is that they form cross bridges. They bind to the thin filaments. Forms a cross bridge. It looks like a drawbridge that's been lowered down. These are cross bridges. We'll see why that's important here in a minute. So thick filaments made of myosin. They have myosin heads that can grab onto the actin. Grab onto the thin filaments made of actin. You need something to stabilize these thick filaments. So you can see attached here to the ends of the thick filament and over here on either end. It's a protein called chitin. Sounds like titan, T-I-T-A-N. Chitin means big. This is the largest protein in the body. Fun fact. I'm not going to test you on that, but it's called chitin. That's why it's called chitin. It's a protein that helps stabilize and provide elasticity to myosin as it's moving. So myosin is the real important player here. Chitin is an important stabilizing protein. So that's thick filaments. Focusing on the thin filaments, if we look at what's going on here, whoops. What we really see is that actin is made up of a bunch of smaller molecules. All these really tiny, small circles you see, these spheres, those are the actin proteins. It has a specific name called G-actin. I'm not going to test you on that, but these are the actin proteins. To help keep these actin proteins in place, just like how we had chitin for myosin, you have a kind of a skewer protein in the middle called nebulin. Nebulin keeps the actin proteins in place to make the majority of the thin filament. Running along, if you can see my blue, running along in a helical way, running along the length of the thin filament is another protein. So we've talked about myosin, we've talked about chitin, actin, nebulin. Here's another protein. It's called tropomyosin. 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 We'll get to what it does here in a second. And the other protein that interacts with it, which I'll draw in red. We have another protein here called troponin. Troponin holds tropomyosin in place. So it keeps tropomyosin in a certain spot along the thin filament. When troponin keeps tropomyosin in place, tropomyosin covers up the spot that myosin wants to grab. I'll say that again. When troponin holds tropomyosin in place, tropomyosin covers up a spot on actin. That spot on actin is the same spot that myosin wants to grab. So effectively, troponin and tropomyosin are preventing myosin from binding to actin. A lot of protein interactions here. Why do we have all that happening? Here is an animation from Crash Course, really useful if you haven't watched this video, of what's going on. For a contraction to happen, we need these proteins to work together. For them to work, here's the first step for contraction. When contraction happens, for a contraction to happen, you get a release of calcium ion, from the sarcoplasmic reticulum into the sarcoplasm. So calcium ion is here in the sarcoplasm. This is calcium ion. And calcium ion binds to troponin. When calcium ion binds to troponin, tropomyosin moves out of the way. So it used to be in one spot and it moved to a different spot. When calcium binds to troponin, tropomyosin shifts. Because it shifted, now there are these binding sites on actin that are now exposed. They were once covered up, but now they're exposed because tropomyosin has shifted out of the way. This is our thin filament. This is our thick filament down here. You see the myosin heads? Myosin heads want to grab onto actin, but they can't with tropomyosin in the way, unless calcium binds to troponin and moves tropomyosin out the way. And so guess what happens? Myosin can grab and pull on actin. Myosin grabs and pulls on actin. This is contraction, the beginning of contraction. It's called a cross bridge cycle because we're forming a cross bridge. We're forming a cross bridge here. When myosin grabs onto actin, that's how contraction happens. And this is just showing one myosin head grabbing onto actin. You have to imagine this happening with many myosin heads, and they're grabbing and pulling, grabbing and pulling, grabbing and pulling. Remember that example of the rowers? They pull back and stroke, pull back and stroke. That's what's happening here. When you grab and pull on something, you have to reset over and over, grab and pull, grab and pull. All right. Back at what's going on here in this molecular anatomy. Shown here, this distance is one sarcomere. How do we know what the definition of a sarcomere is, like the borders, the boundaries? On one end, you'll have these lines that are holding the thin filaments. This is called the Z line. Z line holds the thin filaments in place. What they're also useful for, which is not pictured here, but I'm going to draw it in, is that titin connects the myosin to the Z lines. So I'm drawing in titin now. Wherever you only find thin filament, where there's no overlap with thick filament. So for example, you can see thin filament here. There's thin filament, what I just traced over. There's thin filament here too. But this part is overlapping with thick filament, whereas this part is not overlapping with thick filament. The part that does not overlap with thick filament That's known as the I band. Should use a different color. The part that does not overlap with thick filament, where you only find thin filament, that's called the I band. You can see this with a transmission electron microscope. Here's the I band. You can even see the Z line, that darker line here in the middle. That's the I band. Wrong color. There we go. Okay. I band, the letter I stands for isotropic, means uniform in terms of light patterns. If that doesn't stick with you, I is for light. There's an I in light. It's a lighter colored band. The darker bands where you do find myosin, there is overlap, but where you do find myosin, those are called the A bands. A stands for anisotropic, which means polarized. If that term does not stick with you, there's an A in dark. There's an I in light, there's an A in dark. So the A band is dark, the I band is light, and you get a repeating pattern of this over and over and over again because you got a bunch of myofibrils stacked on one another, and you've got tons of sarcomeres along those myofibrils. Now there is a region where it does overlap between actin and myosin. And that's called the zone of overlap. Getting messy here. That zone of overlap you can see because it appears darker with a transmission electron microscope. You cannot see this with a light microscope. You can see the zone of overlap. So within the A band, let me make that clear. Within the A band, you have zone of overlap. You have an area that looks lighter colored. Do you see the lighter color here in the middle? Well, not in the very, very middle, but on either side of the middle. That is known as the H zone. H for heller in German. Heller means brighter. H zone. And then the very, very middle, the very, very middle is the M line. Just like how there's a Z line between the thin filaments, there's an M line in the middle of the thick filaments. M is for the middle of the sarcomere. So let's summarize at the top. Now, why do we bother naming all these weird lettered regions of a sarcomere? Here's why. Think about what would happen with contraction. These myosin heads grab on to the actin, and when they grab on them, they pull towards the M line. They grab and pull towards the M line on either side. So this is going to move towards the middle. This is going to move towards the middle of the sarcomere in either direction. And that's going to shorten the whole muscle overall. So we can say what happens to all these different things when contraction happens. Well, first, the term for all of this, for when we go from a relaxed muscle to a contracted muscle, this is called the sliding filament theory. The word theory in science means a well-supported, well-evidenced idea. That can still change with more evidence, but this is our best understanding of it now. It's not some weird crackpot thing. It's very well studied, but still, who knows, we could learn more. Anyways, sliding filament theory, proteins are sliding across each other. Usually when you contract something, like when you have the word cannot, and you contract that word to can't, you've literally removed letters to form a new word. We're not removing protein, we're not removing muscle, we're overlapping them. That allows you to slide back and forth, back and forth, back and forth, which is what we need to do. So these proteins are sliding over one another. We're not losing protein. The thin filaments are sliding over the thick filaments towards the M line. Here's our M line. They're sliding to the middle. When the sarcomere shortens, the I band, we're losing I band because it's going to overlap more. The I band will shorten. The H zone, you're going to get more overlap, so you're losing H zone. H zone shortens. You can see that. I band shortens. H zone shortens. There's going to be more overlap, so we go from a small overlap, a small amount of overlap, to a larger amount of overlap. That makes sense because the actin and the myosin are interacting more. The Z lines start wider and they move towards the M line. They get closer. The two things that remain constant based on our frame of reference, the M line stays put. That's our middle. Everything's moving, but we're calling this the middle and it's not moving. And then the A band overall, because the A band is irrespective of overlap, it's just where the thick filaments are. Thick filaments aren't changing, so A band stays the same. A band remains constant. These are changes that we see during contraction, and then relaxation would just be the reverse. You go back to original state. So, as you can see, there's a lot of organization here in a muscle. Whole muscle surrounded by epimysium. Whole muscle is made up of fascicles, which are surrounded by paramysium. Fascicles are filled with fibers. Fibers are surrounded by endomysium. Fibers, the outer covering of a fiber is sarcolemma. And within that sarcolemma, you've got myofibrils. Myofibrils are surrounded by sarcoplasmic reticulum and T-tubules. In the next video, we'll see how T-tubules spread electricity, which triggers the sarcoplasmic reticulum to release calcium. Calcium signals to troponin, which moves tropomyosin, which allows myosin to bind to actin. And the thin filaments slide over the thick filaments within a single sarcomere, causing a sarcomere to shorten. Rewind that again if you need to hear it again. That was a lot. We'll see more in the next video. So let me know if you have more questions. Thanks for watching. Yeah, let me know if you have more questions.